All right. So today's lecture is going to be primarily focused on the extracellular matrix. And what's going to be a little bit different about today's lecture is rather than focusing on a disease as the clinical application or clinical wrapper, I'm going to use this opportunity to instead talk about an entire field of research that tries to leverage the extracellular matrix. And that's going to be tissue engineering and regenerative medicine. So what are tissue engineering and regenerative medicine? Those two fields are sometimes, those two terms are sometimes used interchangeably. Did you have a question already? Okay. They're used interchangeably, but they can mean specific things. So tissue engineering in particular evolved out of the field of biomaterials. Biomaterials was historically a field focused on trying to develop implants that induced very little responses from the body. They wanted to be bio-inert. However, back in 1993, there was a study published in Nature from the group out of MIT, Bob Langer's lab. Bob Langer, who was the pioneer for the entire field of tissue engineering, is also the founder of Moderna. And so he, back in 1993, decided that they wanted to try and figure out if there was a way to develop implants that could be used to replace tissue. Tissue engineering also, as a field, was born out of insufficient donor organs available or tissue donors available. And so they wanted to figure out, can we engineer human tissue using biomaterials? And so what they did was they created a structure that resembled a human ear, implanted it in the back of a nude immunodeficient mouse, and demonstrated that by simply just having the structure, the scaffolding, they were able to leverage the biology of a mouse and have the cells populate the scaffold to create what resembled an ear. And so the field of tissue engineering tries to take a few pieces together to create some of these different therapies. So typically they'll combine scaffolds with cells or with some other biologically active material to try and create functional tissues. There's a professor down at Wake Forest, Anthony Italo. He literally has these little small reactors to try and grow entire human organs, to try and grow entire bladders or entire kidneys. As you can imagine, the more complex or complicated an organ structure is, the harder it is to try and recreate it. So far up to this point, artificial skin and cartilage are examples of engineered tissues that have been approved by the FDA. However, there has been very limited use in human patients. When you have multiple things coming together, like cells, some sort of scaffold implant, and cells, scaffolds, and some sort of biological drug or protein, there's different regulatory hurdles for each of those, and combine them all together, the regulatory hurdles can increase dramatically. But as a field, tissue engineering has a lot of promise. So how is regenerative medicine different from tissue engineering? Regenerative medicine as a field is just a broad field that includes tissue engineering within it, but rather is a field that tries to leverage and incorporate and it tries to leverage self-healing properties that exist already, or tries to promote healing where otherwise doesn't occur. So some examples of self-healing that already occurs is typically with partial liver donors, it leverages the fact that the liver can regenerate itself if only part of the liver is resected away. Other examples of self-healing are bone. So if there's some sort of bone fracture or defect in the bone within a certain distance, the bone can remodel and grow back and reattach. But there are other tissues like nerve tissue that will typically not have self-healing properties. And so there's a lot of efforts in regenerative medicine to try and either accelerate healing or promote healing and regeneration where it doesn't already occur. What's key to know is that both regenerative medicine, but in particular tissue engineering, those scaffolds that are used try to leverage and mimic the extracellular matrix. And so a broad definition of the extracellular matrix is that it's a combination of proteins and polysaccharides or repeating sugar units that are locally secreted in the milieu or environment of cells, and it assembles into this matrix or mesh that kind of creates the environment that cells live in. There's two main classifications for the extracellular matrix. One classification is the basal lamina, also called, that's kind of the antiquated term, more commonly referred to as the basement membrane. The other classification is the connective tissue. So for the basement membrane, typically what happens is there is a sheet of many cells together that are lined up, and underneath them is a basement membrane. And this exists across a lot of different in a lot of different tissue contexts, but in particular that exists under a sheet of epithelium or under a sheet of endothelium, the cells that make up the inner lining of our blood vessels. There's an electron micrograph at the bottom right here where there are epithelial cells on top. Right underneath that is this really thin sheet of basement membrane. And typically the basement membrane is about 40 to 120 nanometers thick. And so the other example of the extracellular matrix is the connective tissue. And this is one that I'm biased because this is kind of part of what I did for my postdoc research. But most people that I talk to when we talk about the extracellular matrix, this is the connective tissue is usually what comes to mind. And there's different, when you look at the basement membrane versus connective tissue, you can appreciate there's a different ratio of cells to extracellular matrix, or another way to call the extracellular matrix is the ECM. So with basement membrane, there is a lot of cells in a very thin sheet of ECM. In connective tissue, there are a few cells that exist there's a fewer number of cells that exist within it, as depicted in the top right on the slide. But those few cells exist with this entire matrix all around them that make up their environment. And within that there can be lots of different, there can be a heterogeneous population of cells that exist together. Bless you. So the physiological roles of the ECM, particularly for connective tissue, it provides scaffolding. It provides structure and shape. When you think about what is all the connective tissue throughout our body and across different tissue organs, that's the ECM providing scaffolding and shape to those tissues. It also provides tensile strength. So in particular, one protein, collagen, is incredibly important based on its structure, and we'll talk about it later on today, and its ability to provide tensile strength. And if you take a moment to put your pens down or step away from your keyboards, our skin has, as you can imagine, has a lot of collagen in it, but it's also oriented in specific directions. And these directions are called Langer lines. So if you imagine, it's easy to pull on your skin here, but because in our forearm, the ECM is kind of oriented this way, if you try and pull here, it's a lot harder. Just take a second to do that. It's easy to do it here because it's the alignment of our ECM, but if you do it here, it's a lot harder and not as easy. And so that part of the reason why is because of the way that our ECM is oriented in our skin. Another example is cushioning. The cartilage in our knees that will eventually degrade away as we all get older provides cushioning and support to enable us to move, run, and jump without too much pain, so that we don't have bone-on-bone action in our joints. It can also serve as a molecular filter. When we have two sheets of basement membrane, they can serve as a filter. For example, in our kidneys, the glomerulus can serve as a filter through what's present from what's in our blood that's going to be filtered out into the kidney. It can also serve as a boundary across different tissue types. A storage depot. This is an active area of research where growth factors can bind to the ECM, but then are released in certain conditions. And also the conformational blocking of cryptic sites, which we'll talk about later. Those are structural roles. The biological roles, which are, I think, really interesting. They can provide cell polarity. This means directionality of the cell. So if I go back one slide, on the basement membrane image to the bottom left here, because of the basement membrane here, provides cues to the cells on top of them because the basement membrane is here, this is your bottom and this is your top, or this is the apical and basal part of a cell. And so that it provides cues for where a cell is and what direction it's in. It can promote cell adhesion, binding to certain structure. And typically what's really important is that a lot of cells adhere to their environment. When they don't have adhesion to something, that actually can promote apoptosis. And again, apoptosis is programmed cell death. And so it's important to know that the ECM, particularly the basal membrane, provides anti-apoptosis cues to cells that are on them. Other important cues are morphogenesis and differentiation. Morphogenesis is a way to describe morphogenesis is it's the development of new tissue, typically during growth and development. Differentiation is the specialization. And so what ends up happening is that if you take stem cells and put them on a basal membrane from bone, those stem cells will become bone. If you take those same stem cells and put them on ECM from blood vessels, they'll start to become, they'll differentiate and specialize and become associated with blood vessels. And so the ECM provides cues for cells to become differentiated or more specialized. It also promotes cues for migration, proliferation, and I also talked about apoptosis. A lot of these proteins are really critical for life. And so as early as a 2-day-old blastocyst, laminin, one of the ECM proteins, is already expressed. Usually what happens is in genetically engineered mice, if we knock out one of those genes for one of these proteins, and this table to the right is a list of what happens when each of these specific proteins are genetically knocked out or deleted in mice. What you'll notice is that for a lot of them, it is either lethal or leads to a really bad disease outcome that it promotes. And so all of these ECM proteins are incredibly important for normal biology. So typically in the ECM, we have two main biomolecules here. We have fibrous proteins that we call glycoproteins. I'm going to take a second to write this out really quickly because I think it'll help. So we have glycoproteins. And we also have these repeating sugar units called glycosaminoglycans, or also called GAGs. When GAGs are associated with some protein, those are called proteoglycans. So for a long time, I can remember being really confused as an undergrad and trying to determine the difference between glycoproteins and proteoglycans, because it feels like it's the same terms, just kind of switched around. But what ends up happening is that glycoproteins are mainly proteins with a very small amount of sugar molecules on them, whereas proteoglycans are a small amount of protein as like the core protein that is covered in GAGs, covered in glycosaminoglycans. And so what that means is proteoglycans by weight are mostly GAGs and glycoproteins are mostly protein. So to remember which one is what the ratio is or which one is which, look at the suffix. Glycoproteins are mostly proteins. Proteoglycans are mostly going to be GAGs or glycans. They're going to be mostly sugar. And so a way to try and tell the difference between the two is to look at the suffix to try and remind you what that molecule mostly consists of. And so we have, so glycosaminoglycans, one example of it is heparan sulfate to the top right here. They are characterized by having high saccharide units, repeating 2 sugar molecules there. And their length is dependent on what kind of species of GAGs they are. And on the bottom left here, we have images of different proteoglycans and glycoproteins. So the green denotes protein and the purple denotes GAGs here. So you can see that laminin, type 4 collagen, nitrogen, and fibrillar collagen, as well as fibronectin, are mostly going to be, those are glycoproteins that have mostly just proteins on them. We have perlecan here, which is a glycoprotein as well. Hyaluronin, which is a GAG that has almost entirely, which is all just glycosaminoglycans. Aggrecan is has a core protein that is covered in GAGs. And so a lot of proteins on average are about 50 kilodaltons. Aggrecan, because of how many GAGs it has on it, is 3 megadaltons in weight. And so what you can appreciate again is in the bottom right here, another image that shows the difference between glycoproteins, what they look like versus a proteoglycan. Before I move on, are there any questions so far? Yeah.
So now what we were just saying, the proteoglycans, how do you relate those to the GAGs? The GAGs are part of it, but they have sugars and.
So GAGs are the sugars on proteoglycans. That's a good question. So just to repeat that again, the way GAGs and proteoglycans are related to one another is that GAGs are the sugars. They are the GAGs are the sugars on proteoglycans. So looking at aggrecan here for a moment, this is a core protein that is covered in glycosaminoglycans all over, and GAG molecules all over. Yes. Both are true. You can have just GAGs. For example, we have hyaluronin over there. Or you can have GAGs with a core protein, which would be a proteoglycan. It's a GAG that has many repeating disaccharide units. Another GAG example is up top, heparin sulfate. It just has a different set of repeating disaccharides and different bonds between them. Yes. All right. So we're first going to take a diver deep looking at the basement membrane a little bit. So as I repeated before, the basement membrane is a version of the ECM that underlies sheets of cells. One common sheet of cells that it typically lies underneath is our epithelium. Many of our mucosal layers throughout our body have an epithelium surface that underneath it has a basement membrane. And so that's what's illustrated in the top right here. It can also surround cells. And so in muscle tissue, as shown in the bottom right here, you can have a basement membrane that wraps around muscle fibers. And so it can underlie sheets of cells or surround cells as well. And another version of basement membrane can also be between cell sheets. So an example, again, is the glomerulus within the kidney. Between endothelium and epithelium, we have a basement membrane that serves as almost like as a filter between the two of them. The basement membrane has four components, but there's two of them in particular that I want you to remember. Basement membrane, the two key proteins are laminin and collagen 4. What ends up happening is that both laminin and collagen 4 are secreted by cells and laid down underneath them. And what ends up happening is that there are two other components, nitrogen and perlecan, that bridge and cross-link those two sheets of laminin and collagen 4 together. And so let me repeat that again. So typically the basement membrane is largely laminin and largely collagen 4. And there are two sheets that are secreted by cells that kind of assemble together, that's manipulated by cells. But these two sheets are cross-linked and bridged together by the other two components listed, nitrogen and perlecan. And typically, again, the thickness of a basement membrane is about 40 to 120 nanometers thick. So to take a second and look at laminin. So laminin is a heterotrimer. That means it is a mixture of three different subunits. The alpha, beta, and gamma chains. Now I know I just said subunits and then I said chains. So when we talk about light and heavy chain, that's typically how proteins separate via enzymatic digestion and how they separate when we look at them experimentally. In this case, alpha, beta, and gamma chains are distinct subunits that are encoded by distinct genes within our genome. There are 5 different alpha subunits, 3 different beta subunits, and 3 different kinds of gamma subunits. And so the different combinations of those different alpha, betas, and gammas leads to about 16 different isoforms of laminin identified in nature. Laminin 1, which is also called laminin 111, which means it has the alpha 1 chain, the beta 1 chain, and the gamma 1 chain, is the most predominant isoform of laminin in our basement membranes. And laminin is key because it helps to organize our basement membrane. And we'll talk about soon about collagen's key role. the key structures to consider for laminin, it has these two short arms, the beta chain and the gamma chain. The beta and gamma chains are key because they are important for self-assembly, where these short arms are where adjacent laminin molecules will bind to each other. And so if we were zooming up on laminin in a basement membrane, we'd see another laminin molecule here with them connected via their beta and gamma chains, and as well as on top. And what I forgot to show is this is what the different isoforms of laminin look like, and because they're different subunits that they possess, that can also affect differences in their. But by and large, as depicted by laminin 1 above, it's usually a cross-shaped molecule, as you can see here, based on old rotary. I feel bad saying old, but old rotary shadowing techniques. We have newer techniques that help us appreciate and see a finer structure of what laminin looks like. I tried to quickly find one and introduce it, but I'll have subsequent versions of the class have that. But what's key to know is that the long arms, the alpha chain here, are really key for cell binding domains. So in particular, the C-terminus end here is really adept at binding to integrins and having changes that can regulate what kind of integrins will bind to it. We usually have 24 different kinds of integrins. And so both ends, both the N and the C-terminus, but particularly the C-terminus end of the alpha chain is key for integrin binding. Collagen type 4 is the other key protein in our basement membrane. Laminin accounts for about 50% of the basement membrane protein, and laminin accounts for 50%. Collagen type 4 accounts for about 50%. And this protein consists of 3 alpha chains. And so they form a triple helix structure together, where most of the entire length of collagen type 4 is characterized by having these 3 amino acid motif repeats, where it's glycine, 1 amino acid, then a different amino acid, repeating over and over and over. There's one particular amino acid that is present as one of the amino acid repeats here. And as evidenced by, I'll give a hint, by the kinking and turning of collagen type 4. Does anyone want to venture a guess what amino acid is key there? Proline, exactly right. Proline is typically one of the amino acids in this 3 amino acid repeat. There's also oftentimes another form of proline that's present called hydroxyproline, but we'll talk later about what exactly is hydroxyproline. I'll take a second for this to come back. But in particular for collagen type 4 that's present in our basement membrane, this is a form of collagen. There are I think as many as 25 or 30 different types of collagen that have been identified so far. But in particular, collagen type 4 is key for the basement membrane and is more flexible, as you can imagine, where if there are sheets of cells in our mucosal linings that need to move, it would be helpful if it was more flexible, as opposed to another form of collagen that was super rigid and provided incredible amounts of tensile strength, like perhaps collagen that's around our aorta that tries to withstand lots of pulsatile flow, being right outside of our left ventricle, where huge amounts of pressure are present. But even with that being said, collagen type 4 provides a lot of tensile strength to our basement membrane, while it being a form of collagen that's more flexible than its other collagen counterparts. Nitogen is a glycoprotein that has 2 isoforms, nitogen 1 and 2. And again, as I said before, it's one of its key function is bridging the sheet of collagen type 4 and collagen type 4 and laminin, bridging and cross-linking those 2 sheets together. Perlecan has the gag of heparin sulfate, and it's a proteoglycan that, again, as with nitrogen, tries to bridge the two sheets of collagen type 4 and laminin together, but it can also be found within our connective tissue. Before I go on, I'm going to stop and give space for a couple of questions here. Yes.
So why does it have to be cross-linked? Why can't it just be like a bilayer? Is there an important...
If you imagine, so think of, what's a good way to describe this? You know, it's really key for if this is providing, if part of what it's doing is providing structure and stability, if these two sheets that are present aren't combined together, that's going to really jeopardize or compromise the stability of that basement membrane lining.
But you said it was like almost 50% and 50% of those two main ones making just the plates. So
right, but if they're not cross-linked together, because what's the way to describe?
Cells linked together, or is it just through the other two?
So the two sheets of collagen type 4 and laminin are cross-linked together. Having that stability enables a whole sheet of cells to be present on it and not have these two sheets sliding past each other or disassociating, but rather these nitrogen and perlecan kind of create these like stability points where it's not going to disassociate or fall apart, if that makes sense. If you can imagine, like if there's two rows of students here, and if I wanted to try and, you know, separate you two, or try and, if there was like a few other people up here trying to pull you guys around, you guys would be a lot stronger if you guys were linking arms across the two of you. It'd be a much more stable structure, for lack of a better phrase.
Yeah.
Are there any other questions? All right, so the assembly of our basement membrane. So typically what happens is these glycoproteins are assembled within our Golgi. We're going to talk about further throughout the semester what the Golgi does, but right now that's a key organelle that's part of protein synthesis. And so here what ends up happening is within the Golgi, both laminin and collagen type 4 are synthesized in their pro-protein form. We've talked about this already, but pro-proteins are the like precursor or immature form of a protein, where they have a pro cap on them. This is pretty key because this pro cap stops collagen type 4 and laminin from creating a sheet inside our Golgi. So we talked about before how laminin on the short arms will want to assemble together with other laminins, but these pro caps stop that self-assembly within the Golgi. And once they're secreted, there are these enzymes on our cell surface. Give me one moment. Here we go. As they're secreted from the Golgi into a secretory vesicle and released out, there are enzymes on the surface of our cells that will actually cleave off the pro cap and also our integrins that bind to these ECM proteins will actually start to manipulate them and lay them down. And then now that they don't have the pro cap, they can actually start self-assembling together to create this sheet. And again, we have nitrogen and perlecan, which will cross-link and bridge these together. So again, the key take-home message here is that within our Golgi, laminin and collagen type 4 are synthesized, made with a pro cap on them. So they're in their pro-protein immature form. When they're released or secreted, the pro cap is cleaved by enzymes on our cell surface, and then the cell will begin to lay down collagen type 4 and laminin, which will then self-assemble together to create this network or this sheet, which will then be cross-linked and bridged together by nitrogen and perlecan. I just scooped myself. It's crossing together by nitrogen and perlecan here. And so again, as I was mentioning earlier in response to a question, the nitrogen and perlecan, if these two sheets are laid down, unless they're cross-linked together, that's going to really compromise the stability of that basement membrane. So we talked a little bit earlier about cryptic sites on an early table for like what are like physiological roles of the ECM. Cryptic sites are typically hidden sites that when they're revealed will elicit a different biological response. So an example of cryptic sites is with laminin during tissue regeneration. And so what ends up happening here is that when laminin, when this form of laminin binds to integrins on its C terminus here, what ends up happening is that during one phase of wound healing, these LG modules or laminin globular modules, we have 1 through 5 here, they'll typically bind one form of integrin, alpha 6 beta 1. Every integrin has an alpha version and a beta version. We'll get to like what those are a little bit later, but just all you need to know is that integrins are receptors for the ECM, and one kind is alpha 6 beta 1. If we lose these two LG modules, laminin globular modules 4 and 5, that reveals a new cryptic site where now a brand new integrin will bind to them, which induces a totally different physiological response during the wound healing cascade. And so typically alpha 6 beta 1 is pretty important during migration, and alpha 6 beta 4 is important for forming cell-to-cell contacts between cells. And so they kind of help direct like epithelium regrowing during the wound healing response. So that was a lot of information, but the key take-home message here is that these ECM proteins have cryptic sites where there's these regions of them that are hidden, that when they are made available, in this case, plasmin is an enzyme that cleaves off some of those LG modules, these cryptic sites become available and elicit a totally different response. That's the key take-home message here for this slide. Next is the connective tissue. Before I move on, I want to give space for a couple of questions here. Does anyone have any questions? Yes. So the integrin will bind, like this is like the integrin binding site here, but then the enzyme, in this case it's plasmin, will cleave off these two modules. And so like integrin's bound to those, but since it's not really inducing any response, it'll typically be recycled and that won't be an issue anymore. But there's this new binding site that's made available that will bind to a different integrin. So integrins are the receptors on our cells for these ECM proteins, and different integrins elicit different responses in our cells. This is kind of like looking ahead to future content in this class. But again, the key take-home message is this is an example of what I'd like to do during office hours. Rather than focusing on the different LG modules, the different integrins that are bound, what those different integrins do, what I want to draw your attention to is that on our ECM proteins, there is like one version of it that is like available to our cells that induces a response, but then sometimes there's like a cryptic site that is hidden that becomes available that induces a totally different response. So these cryptic sites can be important for regeneration, can be important in disease. Some forms of scarring might be triggered by cryptic sites that become available that makes cells think, oh, this is a scar tissue, let's make more scar and more scar, even though there was no scar to begin with. So like this is an active area of research. Like there's like a few labs down the hall right now that are answering questions about cryptic sites and how they're regulated and how we can create biomarkers to identify them. But again, the main take-home message is that these cryptic sites exist on our ECM proteins and just elicit different reactions. Mm-hmm.
Does every ECM protein?
Great question. We're still learning that. Or rather, perhaps how many they might have. There's another, so like Dr. Thomas Barker here in the department, his ECM protein that's near and dear to his heart is fibronectin, and he's spent the last 10 years kind of like going through, 10-ish years, kind of going through and identifying different cryptic sites on fibronectin, and now like making antibodies that can serve as a drug to identify them and also to block them as well. And so that is a whole entire area of research. We don't know all of them, but you can go get a PhD and find out a new one. All right, I'm going to press on to try and get a couple more slides in before we run out of time. So we spent time talking about the basement membrane. The other form of the ECM is the connective tissue. So again, it's important to keep in mind that the basement membrane, the ratio of cells to protein or cells to ECM, excuse me, is that for the basement membrane, it's a lot of cells, typically, with the exception of filtration perhaps, but a lot of cells with a small, thin layer. For connective tissue, what we have is a few cells in a huge gelatinous structure of ECM proteins. And so it's typically beneath the basement membrane, or it can also be around the basement membrane. So in the case of muscle, where there's a basement membrane around cells, around that basement membrane is a gelatinous connective tissue ECM all around it. And so typically, the components of our connective tissue ECM are protein fibers that are within a large gelatinous structure of like proteoglycans. And it's more again, the ratio, it's more of ECM than is to cells. And this is a key feature that determines our tissue's physical properties, how stiff it is, how pliable it is, how rigid it is. And so it's, I think, more interesting and compelling than basement membrane, but we can differ on that. The key cell that is important for the production of the ECM are fibroblasts. They are the predominant cell type in connective tissues, and there are different species of fibroblasts across different tissues. They might go by different names, but they're all throughout all of our tissues in our body. Fibroblasts are what secrete most of our ECM. They secrete the fibrous proteins that provide strength. They also secrete the GAGs and the proteoglycans that have immense negative charges, and we'll talk about, hopefully next class, we'll talk about why those negative charges are important. And they're incredibly hydrophilic. They can contribute to the hydration within our different tissues, and they provide resistance to compressive forces. I'm going to hit stop there, and we'll pick up where we left off next class. Thank you.